40 years ago, a microbiologist at UC Davis found a DNA fragment inside a soil bacterium that had no business being there. He published it. That molecule is now being engineered as a write head for one of the densest recording media in existence. DNA can theoretically store 215 petabytes per gram. In July 2024, Gonzalez Delgado, Lopez, and Rojas Montero at the Gladstone Institutes published in Nature Chemical Biology, Retron arrays can perform simultaneous multi-site editing within individual bacterial genomes, a step toward combinatorial event logging. Proof of concept recording in mammalian cell lines has been demonstrated, though reliable in vivo tissue recording remains early stage. That last detail, no external template delivery required per event, is the technical wall most competing approaches hit, and it's why researchers see Retrons as a potentially unique substrate for in vivo recording. Seth Shipman trained as a neuroscientist. His problem: by the time you can measure what a living brain has done, the moment that made it is gone. A molecular recorder embedded in the cell itself could change that, if the engineering gaps close. Here's how the recording works in current cell line experiments. A stimulus, a drug, a stress signal, a tumor, suppressor failing, triggers the Retron's reverse transcriptase, which writes a heritable barcode into the genome readable by sequencing weeks later. Track direct funding sits at roughly $3 million, but adjacent programs, DNA typewriter, Ngram, CRISPR recorders, put estimated cumulative global investment exposure between $340 and $480 million through 2026, according to analyst estimates. The Shendur Lab at the University of Washington is building a parallel architecture called DNA typewriter, and their July 2024 Ngram system recorded nearly 100 transcription factor activities using prime editing rather than Retrons, but converging on the same goal. The catch is real. Editing efficiency in mammalian cells currently sits at just 1 to 10% per event. Cargo is limited to roughly 150 nucleotides, and cell-to-cell variability creates noise that confounds longitudinal interpreting.